Okay, hello today. I don't know if you can see me real well, but for some reason it looks really dark, but this is me. I'm Jen Durkitz. I'm not sure why the camera's so dark, but you should be able to see my screen, and I'm actually in your class, so I am Professor Durkitz, the woman behind the mystery. Kind of like the Wizard of Oz, hey? So I hope your semester is going well, and I want to reiterate that if you have questions on particularly the labs, please let me know. And also Professor Neal and I will be both on the Collaborate session sometimes, maybe just me, maybe just her. So anyways, I wanted to do a little video on your BLAST, which is week 5 lab. And so I'm actually in your class, and you'll see I'm going to, now this is my view, so keep that in mind when you're looking at it. So I'm going to click on lab. And I'm going to scroll down. And you can see here that I'm just talking about the BLAST search for online. There's no class meeting this week coming up. Okay, so we're going to scroll down. Just wanted to point that out. And we keep scrolling down. Week 1, week 2, week 3, week 5. Okay. So I'm going to click on BLAST. And it's looking a little different for some reason. I'm not sure why it looks like this, but I think it might be because I have my screen condensed. Okay, so I wanted to point out a few things that are in here. So here is a paper on BLAST that will explain a little bit more about what BLAST is and give you some information on how powerful this tool is. So this is not the only tool that's out there to search for DNA segments or genes and their alignment. So as you know, DNA makes RNA, RNA makes proteins, and then they go backwards. Okay, so that wasn't always the case, and it wasn't always known. But now that we know we can do that, this site was actually developed as a way of taking a set of unknown or known, whether you had a protein, whether you had a piece of a protein, maybe you just had a gene, maybe you had less than a gene, maybe you have a segment of DNA, maybe you're trying to perform PCR, polymerase chain reaction, on a piece of DNA because you want to make more of it, you want to study it, and so you need to know what primers can be used for it. So this is kind of what you use after something has been sequenced. So let's say that you have the strawberries and peas, okay, and you're doing a DNA extraction. So first you're going to attempt to get the DNA out of whatever organism it is, right? Plant, animal, bacteria, archaea, whatever, okay? Prokaryotic, eukaryotic. You're going to try to get the DNA out, and once you have isolated the DNA, you're going to want to figure out, maybe you're trying to figure out why this strawberry or this pea is bigger or had more seeds on it or something. There's something about it that's different, and you want to know, or in the case of a crime scene, you want to know what kind of DNA it is. Okay, that's where you get into BLAST. And so there's a lot of different applications for BLAST. So when you're writing your lab for this week, you're going to submit your answers to the pre-lab here, okay, but you're not going to necessarily put your pre-lab answers into a report, okay? And then for BLAST, you're going to submit your answers to the BLAST lab here. So you're not going to be submitting a report like for weeks 1 and 2, well, at least for week 1 and 2. Okay, so keep that in mind. That here's where you're going to submit your answers for the pre-lab. So please answer these questions and then paste them, attach them, something in here. and then when you answer your actual lab questions, lab 5, you're going to submit your answers here. Okay. So I'm going to go to the BLAST website, and BLAST stands for Basic Local Alignment Search Tool website, okay? And so I'm going to go there. Okay, and this is what you will see when you get to the website. And please feel free to obviously look around and play with this, okay? So as you can see at the top, you have, and it tells you what it finds. It finds regions of similarity between biological sequences, okay? So you can use, here's nucleotide, right? So this would be your DNA or your RNA. And you can look to see what the nucleotide would be translated into. You can look for protein. So this is like comparing, let's say you had a protein and you wanted to know what kind of protein it was. You know, is it human? What does it do? Etc. You could do that here. That's called a protein blast. And you can also do where you have a protein and you want it to go backwards into the actual DNA RNA sequence. So there's a lot of information. This is a very, very powerful tool. NCBI, the National Library of Medicine, and NIH. So you can actually sign in, but for what we're doing, you really don't need to sign in. You can just search on it. And if you keep going, you can do certain organisms. So you can look at a human organism. So when we talk about like the Human Genome Project, when these genomes are sequenced and people want to be able to study them and you're comparing, right? So we have human, mouse, rat, and microbe. So this would be your bacteria and microbiology over here. And they've just given some common ones, but you can enter a scientific name or a common name. You can enter butterfly or, you know, whatever that you want to. You can download BLAST. This website has changed over the years as technology has changed, you know, using BLAST in the cloud, just different things. And then they've done these specialized searches, which is nice. So remember I was telling you about the primers, so you can find, you can design primers that are specific to whatever DNA that you're trying to actually make more of or amplify, okay? So here's the protein one. Let's say, also when you're dealing with proteins, there are conserved domains that have genes that are highly conserved, meaning they stay put from you know, generation to generation. So this is very helpful in evolution. You're looking for, like you have a tree, you're trying to do a tree chart. Maybe you're working with immunology. You can look for immunoglobulins. You can look up gene expression profiles. Maybe you're trying to make a vector, or you're trying to do, like, use a prokaryote and make a bacteria or an E. coli, let's say, and you're trying to actually put a gene that you want to be expressed into it. Maybe there's some kind of contamination that you're dealing with. All kinds of stuff. I mean, there's just all kinds of things that you can do in here. Here's the tree. Like, let's say that you have a marker for a certain gene. You're looking for cystic fibrosis. Maybe you're trying to study it. You're trying to see how far it goes back. You're studying pedigrees. You're studying family tree. I mean, the list is very long. You can look at a targeted loci. Okay, so see, it tells you can search the bacterial, the 16S, and the fungal. Can't get it to come back up again. Well, of course not. But anyway, you can look for lots of different things in here. Okay, so what you're going to do, I just wanted to show you that page because this is a very powerful tool and it's kind of cool to just go in here and play around. So what you're doing in the lab, it'll tell you that this is pretty much what comes up whenever you hit whatever you're looking for, some sort of blast. It's going to look like this. You know, there's the protein blast right there, blast X, blast N, lots of different things. So you can put in, from your lab, you're going to actually, let me go back to your lab here. Hang with me. I'm going to go to your BLAST lab. It's going to come up and it's going to tell you, basically give you more information than you can possibly want to know on it. it's going to show you an electropherogram. There it is. So this is, okay, so let's say that you have extracted your DNA and then it's been sequenced. This is going to show you the actual sequence data. This is what it looks like when it comes off. No, it's not colored, but it gives you a visual representation of the different nucleotides. You can keep going. You can read about that, and then so this is if you're coming into the website from the beginning, and then the BLAST home page. That's where we just were. So that's where we were. Keep going. That's the page that we're on right now. And so I'm just going to show you what this does so that you can do it as well. I don't want it to give me that once pasted thing, but well, that's fine. Okay, so I'm going to copy that out and go back to the BLAST. Where is it? There we are. And I am going to just paste it right in here. Boop, just like so. And it's going to tell me that, you know, if I wanted to specifically do human genome, I could do it there, or mouse, or others. I'm going to leave it on others because we don't know what it's from, and that's what we're trying to find. So if you have something that's unknown, right, you don't want to bias the system. You're going to actually put it in, you're going to say, well, what exactly is this, or what is it close to? It might not give you an exact match because you might not have much of a DNA sequence to go on. You might have half of this, or you might have a lot more than this. You know, you just don't know. It depends on your application or what you're doing. The other thing that you're searching for is you're searching for something that's highly similar. Okay, so we want to give it the best possible chance to come back and say, okay, this is what, you know, based on everything that's in here, what is the most similar? You know, maybe you're looking at something else and you want to know, well, what's somewhat similar, or maybe you know what it is and you want to do a process of elimination. So it just really depends on what you're trying to do. And then you're going to say there's other things that you can go into. You can do algorithm parameters, but for this you're just going to hit BLAST. Excuse me. And it comes up like this, and you have to give it a second. Like, you're going to have to give it a minute. Interestingly enough, it's very nice because it gives you a description. This is kind of new, where it gives you how to read this page. You can download the results. It gives you formatting options, all kinds of stuff. You can edit it and resubmit it. This probably changes every year because they go in and update it. But this is what we're looking at here. This is your query. Okay, so what it has done is it has looked for right here. Okay, these are showing you that there's a score of 80 to 200. So the higher this number, the more similar or the more aligned it is. Okay, so we've got some really strong alignments over here. So that's good. Okay, so that's what that's showing, and to what we gave it. So this is based on what we told it that we wanted it to look for. And so let me show you as it goes down, it gives you the alignments. Okay, so it tells you that the first one, okay, it tells you that with a max score of 363, a total score of 484, there's a 73% query cover. Okay, so that means like over everything that you gave it, it was able to identify using within 73% of it that there was a 99% identity to this accession. Okay, so basically the three highest pieces, okay, 363, 363, 363, okay, and 484, depending on how you want to look at it, tells you that it's Homo sapien, okay, that it's some sort of parathyroid hormone, and that it is found or related to something on chromosome 11. Okay, so just by looking there, you can get a pretty decent amount of information from it. And you keep scrolling down, okay, and you can see that that's not the only one, that the first 6 are Homo sapien, but then it keeps going, well, it could be, oh, you know, here we have gorilla, you know, different things, messenger RNA, which makes sense if you're looking at it from an evolutionary standpoint, right? All right, so we keep going down. You know, here's the actual alignment itself. So it tells you that for this pre-pro parathyroid hormone, that it gives you the actual query. So this is where it's finding what you gave it. Okay, so it's going to show you, it's going to give you some information over here. And if you go to the sequence ID up here and you click on that, it's going to take you into the actual database itself. So what the database is going to do is the database is going to give you whatever information that it has on the pre-pro parathyroid hormone. Okay, it's going to give you the definition of it. It's going to tell you what it is, what organism it comes from. It's going to give you some evolutionary information. So this would be like your phylogenetic tree information. It's going to give you information on papers, PubMed sources, authors. You're going to keep going. It's going to give you some information on the gene itself, like the introns, the CDSs, just peptides, so those are proteins, exons, all the things that you're used to seeing in terms of how DNA are. It's going to show you the actual DNA sequence itself, so you can see that obviously we didn't have like a whole huge set of the gene. And you can, if you go over here, you can get to related information on it. You can find the protein coding sequence, taxonomy, the full text, the gene, all kinds of information that you can see. You can see down here, if I can scroll over, you can see some information on it that it's being used to look for. There are homologs of this gene, which this is some evolutionary information. So this gene is actually conserved, meaning the same, or at least pieces of it are the same, in human, chimpanzee, rhesus, dog, cow, mouse, rat, and chicken. Okay, it tells you what it is, and it gives you some reference sequence information. So this would be, for example, you have the DNA, right, and then you have it makes the primary messenger RNA, and before it goes out of the nucleus, right, it's spliced, and it gets the 5' cap and the tail, and then goes out and tries to find the ribosome. So it really does work very similarly to what what you read. So thankfully, what you read is not is not a joke. Now let's say that you had found this gene and you wanted to pick primers for it. Like, let's say that you had been out in a field, you were studying, maybe there was a crime scene, you thought that it was human, but you needed to make sure that it was human. How do you know it didn't come from a chicken or a monkey or whatever, right? You might want to pick some primers that go along with it, okay? And you might want to design and test primers for that sequence using the primer blast, so that you can do what? So that you can make more. You can amplify it. You can amplify this gene, amplify the sequence that you have, make more, run different tests. Maybe you're going to sequence it again. Maybe you're going to send it down the hall to another lab. They're going to do something with it. So that really is the power of this program. There are other programs that do this out there, but this just happens to be one of my personal favorites. So anyways, whatever information that you're looking for, and sometimes it doesn't give you, like the page that we were just on right here, sometimes if you are up in this region here, you might not get any of these. It just might start with predicted, and so then you're talking about the messenger RNA, right? So you might have to dig a little bit more to find what that is. And it's kind of doing a comparison. That's really what it is. Like, you're looking at these letters, right? And you're saying, well, how many of those letters match something in here? Okay, and we actually do, when you're doing these types of things in the lab, you actually sometimes will get back your sequence data, like that electropharogram. It'll give you a sequence, and you may have to have this piece of paper out, and you may be sitting there trying to figure out where it was homologous or where it wasn't homologous, or vice versa. But as you can see, this is going to save a lot of time at least in terms of big picture, trying to find the identity of something or compare it and confirm the identity of it. It doesn't get down to the nitty gritty, but it can be very, very useful in terms of identification and just learning about something. Like if you wanted to, let's see, we can go to here, see if they'll let me in, and it can come up. So here we have the PMC, and you can come in here and you can find papers on it. Okay, we've got some 1983 papers. So some of these things are going to be older, and some of these things might be newer. But remember, this is basically a collection of what's out there. You can click on, you know, NIH grants if you want. You know, you can see, okay, these information came from grants. Maybe you want to look at something that was from a year, and there might not be anything. Okay, so maybe you want to look for something else. You want to come down here, and you're like, oh, why is this related to it? You know, so there must be something in this paper here that has something to do with the parathyroid hormone. So anyways, you can see how you just can keep digging and digging and digging. Okay, so I'm gonna jump back to lab 5. Okay, so I'm inside your website. So that basically is what you're doing here. So you're going to want to pull, this is the website, this is the actual lab, and the lab, let's see, let me do that again. I don't know why it closed out. Actually, I think it's at the top there. Okay, here we go. So basically what I've done is just gone through the lab with you, and that's essentially what you're doing. You're copying and pasting it, you're blasting it. There it is. It's going to give you some information. It's going to show you all the things that I just did. And then there's some questions about it, and you may have to research a little bit about it to be able to answer these questions completely. So these are the questions that you're going to answer, and then when you're done answering those questions, you're going to plot them right in here. And then the pre-lab questions, let me show you so there's no confusion. Take an hour to come up here. I don't know why it does that, but anyways. Here it is. And so these are just, and you can, you know, this is a Word document, so it's just going to ask you some questions about that pre-lab and blast. And you will take the answers from those questions, and you will plop them right in there. Okay, so these are the 2 submits that I will need to see from you for week 5 blast. Okay, so if there are any questions about this, please don't hesitate to write, and I hope everyone's semester is doing well. And if there are any other questions on anything, let me know. Thanks a lot.